Hi, this is the Chapter 13, Segment 2 outline for behavioral neuroscience. We're going to get started on talking about one of the first ways that the body can try to prevent loss of water volume, loss of water, and thirst. First off, is to know that sodium chloride is actually crucial for fluid balance because it is used to maintain water in extracellular component and therefore needs solutes. Too much salt is known as an isotonic concentration. Seawater is hypertonic, and we actually conserve sodium to conserve water. Aldosterone stimulates the kidneys to conserve sodium. Having too much salt or too many solutes inside the bloodstream is known as osmotic thirst. Getting back to the fact that seawater is hypertonic, we can't drink seawater because it would kill us. It's too highly concentrated with salt. However, certain animals like this bird can have adaptations that allow it to drink salt water. Next, we're going to get into hypovolemic thirst, which is a result of the decrease in overall volume of extracellular fluid. We usually do have moderate loss of volume with everyday life. The big problem would be if you experience sudden and dramatic loss through either a hemorrhage or diarrhea or extreme sweating. Hypovolemic thirst results in a drop in blood pressure because there's not enough liquid to fill up the veins, which causes blood pressure to lessen. However, it doesn't matter how much or how little volume there is, the concentration of the extracellular fluid will not change, because if we lose the volume of water, the amount of salts and nutrients inside the blood will also leave. An initial drop in volume is detected by baroreceptors. And they are involved with the hypertone extracellular fluid, and they can replace water without salts. Baroreceptors detect the drop in volume by actually detecting a drop in blood pressure, and this leads to the heart decreasing the secretion of atrial natriuretic peptide, because atrial natriuretic peptide actually reduces blood pressure and inhibits drinking. At the same time, however, the brain releases vasopressin, which actually constricts the vessels and increases blood pressure. This tells the kidneys to flow water through the bladder. That's for vasopressin to do. A third hormonal response to conserve water is a chain of events in which the kidneys release an enzyme called renin, and this enzyme triggers a hormonal cascade which results in the circulation of angiotensin II hormone. Angiotensin II constricts vessels and also triggers the release of vasopressin and aldosterone. It also regulates behavior through cations at neurocytes in the forebrain, specifically the circumventricular organs. In the circumventricular organs are fenestrated capillaries, which are capillaries that lack a blood-brain barrier. And due to the fact that they don't have a blood-brain barrier, it's actually easier for neurons to monitor hormones. The organum vasculosum of the lamina terminalis and the subfornical organ are prompt large drinking response when angiotensin II is detected. The subfornical organ contains an on-off switch for drinking because it can both stimulate drinking and inhibit it. It uses a combination of signals to monitor how much water we ingested and stop in anticipation of fixing the extracellular volume osmolarity. Because sometimes we can drink more water before our body realizes that we don't need to drink any more water. However, most animals know how much water to drink through experience, because experience teaches us to gauge accurately if we've ingested enough water. Cessation of one thirst, however, will not stop thirst. One thirst signal will not stop thirst. You need the cessation of multiple thirst signals. Continuing on with thirst, thirst is a homeostatic signal that can force itself into consciousness, from subconsciousness into consciousness. And this is through a strong activation of the limbic system. The system for food, now we're going to get into food intake. The system for food intake is more complex than systems for fluid balance and thermoregulation. This is because we not only need food for energy, but actually to also supply us with certain nutrients that we don't make naturally. Our body, for example, cannot make amino acids and a few fatty acids in our diet, and also 15 additional nutrients. Animals can never run out of energy or nutrients. If you would, then we would cease to actually work and be able to maintain homeostasis. So all animals have to have some type of reserve on hand at all times. The reserve can, for example, be in the form of lipids and fat. However, there must be a right balance to where the reserve must be enough to be able to supply us with energy and nutrients if we can't eat, but it can't be too large or it will affect mobility. The energy is gathered when the chemical bonds of complex molecule are broken and smaller and form simpler compounds. To raise body temperature, we release this chemical bond energy from breaking down molecules. 33% of the food energy is used just during digestion of the food, and another 55% is used by our basal metabolism. And finally, only 12% of the energy we get from food is actually used for active behavioral processes. Famous scientist Max Keebler developed an equation to relate energy expenditure to body weight. The problem with Max Keebler's equation is that it's not accurate among individuals within a species. It only works with a whole population of a species. Getting into weight loss, a calorie-reduced diet prompts reduction in metabolic rate. And this eating a little amount, and that would such a little amount for a long time, will lower the metabolic rate to such a low amount that it will actually prevent weight loss due to metabolic adaptation. And it can take years for a person's metabolism to go back to normal. If they spend a super long amount, an extended period of time only eating a few hundred calories a day, their metabolism will lower to such an extent that they will have to keep eating that low amount of food in order to maintain the weight they have. We're going to get into the fact that there is some evidence that reducing calorie intake by 50 to 70% can actually extend average life expectancy. And this is due to the decrease in the basal metabolism. Research in invertebrates' pair of genes for transcription factors that are involved in food deprivation lead to longevity. Gains control hormones and trophic factors for longevity through caloric restriction. Production of the SIRT1 marker for increased longevity is increased when you reduce calories.